Let's get going.
I was supposed to go live on custom live stream. I did it wrong.
Oh, well.
Okay.
Take one of your gold stars away from you, Sandy.
No.
And she'll get two back for fixing it, though.
Right, there you go.
Get going. I was supposed to go live on custom live stream. I did it wrong.
Okay. So it looks like we're live now. And so this is, again, the October 2021 meeting of the Cascade Mycological Society with our distinguished invited guests. And I hope I'm not going to mangle your name too much. Dr. Anna Bazzicalupo. Anna grew up in Naples, Italy, and she did her undergrad and master's degrees in Scotland. She just completed her doctorate at the University of British Columbia in May Burby's lab. And she's interested in the taxonomy and systematics of Russulas of the Pacific Northwest. And as well, her work involves using this complicated genera as an example of a collaboration between mycological societies and academia towards the better understanding of the systematics and taxonomy of our local mushrooms. And in addition, she's been working on the Ben Wu Russula collection from the Burke Museum to barcode the Russulas of the Pacific Northwest. And she'll tell us more about Ben Wu and about this amazing project she's been working on for now, I don't know how many years, but without further ado, Dr. Anna Bazzicalupo. Anna, take it away.
Thank you, Chris. It's so great to see you. And hi, everyone. I'm going to start sharing my screen. All right. So the first thing I wanted to do was acknowledge that I'm in the traditional, ancestral, and unceded territories of the Musqueam people here at UBC in Vancouver, Canada. And I'll be talking to you about this project about Ben Wu's Russula Herbarium, and then I'll talk a little bit about something I've been working on in fungal conservation in Canada as well. Because usually what would happen is that I would we would go on a foray and look at the diversity of your local mycota, but because we're on Zoom, we don't get to do that. So to give you a little bit of a background about me, I have 2 slides to show you. And Chris did a great job at introducing me, but I just wanted to give you a little bit more background. So I come from Naples. This is a picture of me and my mom in Naples in 1990 with Naples won the Italian Cup. And we're both wearing our matching jerseys made with tape on a blue jersey. And my dad is Italian, my mom's American. And after growing up there, I went to university, and that's when I started getting interested in fungi during my master's. In my undergrad, I was in Scotland, and I started working on Russulas. And then I had a job in Germany, but then I moved to Vancouver to do my PhD. And I was there for 6 years. And then I moved to Bozeman, Montana to work on a project on Swillis. And then I'm back in Vancouver working on yeast. And part of the reason why my accent is hard to place is because I pick up, I parrot people all over, everywhere I go. So all these projects have to do with understanding fungal diversity, which is something I am interested in. How did it evolve and how it is maintained? But for this talk, because I want to talk about how we discover and describe diversity and why it's important to preserve species that we discover and describe, I want to start even broader than just fungal diversity. These are pictures of organisms that you may or may not be familiar with, but they show an area of diversity that we have on the planet and organisms that we share the planet with. And some of them we know quite well, like these 2 cats that are very closely related, so closely related that they can mate. any very young person is able to tell you the difference between these two cats based on just on these two pictures. You ask a 3-year-old or a 4-year-old, they'll be able to tell you which is which. And one of the reasons why this is important and why we want to know the difference between these two species has to do with communication. And so I want to give you an example of that in this poster from WWF. What I really love about this poster, there is this picture of this wonderful cat made by Stephen Hogg. And then what I love about it is the economy of it. Nowhere on the picture do you see the name of this cat. And then you have this caption that says there were 3,200, how many now? And you don't need to know anything more than just seeing the picture of the tiger to know that what they're talking about is 3,200 individuals of tigers. And they're asking you how many are there now. Now for a thought experiment, what I'll do is this. When you put this picture and you put it on top of there, what you see is a fungus you should be pretty familiar with. But this question becomes confusing because for many reasons. So what are they talking about? Are they talking about mushroom caps? And if they're talking about mushroom caps, that means that maybe they're, why would that be important? Because multiple mushroom caps could be the same individual. Are they talking about individuals, and how can they tell? Because multiple mushroom caps can be the same individual. And then maybe this is just a proxy and they're talking about different species. So it just becomes confusing because even though this is a very important mushroom that you should all know how to recognize, we don't know enough about it to make a statement like this one, which is ironic because you're way more likely to die eating one of these mushrooms rather than eaten by this tiger or a tiger in North America. Okay, so what I'm going to go over is the first part of the talk is going to be about how fungi can be challenging to identify, and I use Russula as my study organism, and I'll show you why it's difficult to identify. And then I'm going to go over why, paired with the fact that the fungi have some cryptic life stages, makes it very difficult to have conservation efforts that are effective, but not impossible. That doesn't mean we shouldn't try. And all along this journey of describing, categorizing, protecting diversity, I want to talk about the role of mycological communities, by which I mean local mushroom clubs and societies, in understanding this diversity. So this is a picture of Ben Wu. I'm going to talk about his collection of Russulas, his contribution to science, and how important collections are in discovering diversity. I never met Ben Wu, and so I know about him through talking to people from societies like yours, excuse me, and through his collection, through reading his notes. So as a mycologist, he was one of the founding members of the PSMS in Seattle in 1964. He was the first president of the society. He was the first chair of the annual exhibit, and he was the first editor of the club's newsletter. He was also part of the Pacific Northwest Key Council. He was also really involved in his community. So as a Chinese American, he was part of the Commission for Asian American Affairs. We don't have to read all this list, but I just wanted to give you a sense of how involved he was with everything, on top of being his full-time job was being an architect. And this is a lovely picture taken by Adolf Cheska. On top of all that, he collected Russulas, lots of Russulas. And I wish I knew who took this photograph to have a less blurry picture of him, because he looks so happy handling and trying to sort out all these Russulas. So just to tell you a little bit about his collection, which is now at the Burke Museum, it's composed of 1,000 collections of Russula done from 1974 to 2007. Most of them have been are from Washington and Oregon, although there's some in Idaho, British Columbia, and California, and I think Alaska as well. I forgot to write that. And each of the collections, he dried the mushrooms and he stored them at first in his basement, and then after he passed, they were moved to the Burke Museum. So each collection is composed of a dried specimen. So these are little dried mushrooms, a photograph, and a collection sheet. So each of these mushrooms, or these collections, so multiple mushroom per collection sometimes, was described in this collection sheet, where you can see there's a collection number, the date, the location. Then there's some information about the morphology that has to do with the cap, the gills, the stipe, and so on. And then there's some chemical reaction. So he would chop up his mushroom and you would test it in different chemicals. So this is an outstanding collection. And this is a photograph of all 1,000 specimens in boxes. I took this at UBC, and each one of the collections has a little barcode, and it's vouchered in the Burke Museum. Just as a clarification, a voucher is a physical specimen that is officially recorded in the herbarium and has a little unique identifier like a barcode and an accession number. And the specimen itself will have some information about its morphology that's contained in there, but associated with each of the physical specimens, there's information there, sometimes on their habitat, location, and field notes. Often, and most of the time, not as extensive as the Benwu collection. Another clarification, what is the herbarium, is a collection of dried or pickled vouchered specimens. And herbaria preserve information about organism so that you can keep them in for posterity, or so that we can consult what was present in the past and learn about it. And this is a slide from Barbara Thiers at the New York Botanical Garden. She kindly let me share this with all the different herbaria of the world. So in Oregon, what you get is 17 herbaria. So you've got a lot of herbaria. And the one that has the most specimens is the one in Corvallis at OSU. But Eugene seems to have one. And I'd be curious if the society collaborates with them at all. Just as a curiosity, I can't speak for every herbarium, but generally anyone could technically submit to herbarium, especially local mushroom clubs. And the only thing that sometimes herbaria can't do is that they don't have enough space, and so they won't be able to take new collections. But if you were interested in preserving your specimens, you could ask questions of the herbarium, like, do you have a protocol to preserve the specimens? Is there a template that I should fill out to submit specimens to the herbarium, etc.? And then they might have information about local areas that are undersampled, and those would be informative for them. So it would give them an incentive to keep your collections. Herbarium, not the only ways to preserve information. Of course, you can have, so Chris was mentioning that you might have a Bioblitz of some sort. So that's a great way to have a species list, forays, and of course, iNaturalist and Mushroom Observer do this. And all these ways of keeping information about species through time have their pros and cons, and I think they're all great. So to go back to the collection, I said that they have a photograph and they have a collection sheet. And the first thing I did for the project was to take the information in the collection sheet, which was only scanned at the time, and make a database that you can actually look up right now if you're on your computer. Just Google Ben Wu's Russula mushrooms, and you can search the collection based on the morphology. So if you wanted to know, give me all the specimens that turn blue in, I don't know, a chemical that he used, like ferric sulfate, you could do that. You would click on this tab and then search that. So that's the first thing I said we because I actually organized a Russula Jamboree with the Vancouver Mycological Society here. And they came and had lunch at UBC, and we set up a bunch of laptops, and they helped me enter the data into the database, which was, I'm so grateful they helped me do that. And then, so we have the database, and then the second thing, the second part of the project had to do with the physical specimen. And so with the physical specimen, what I did was you take a little chunk of the gill and of the Russula. You can extract the DNA and sequence one of the fragments. And so without going into the technical part of that, this is a cartoon of what you would see. So you can see that there's a string of A, T, C, and Gs, and that's what we will call the DNA fragment. And so I had all these Russulas, all 1,000 Russulas, and for all of them, I sequenced the same fragment of DNA. And what you can do with the same fragment of DNA is to compare it. And so imagine you have these Russulas, and you have all the DNA to compare it. You say, okay, at this position, they all have T's. At the second position, these 1 through 4 have a T, 5 and 6 have a G, and 7 and 8 have an A, and then so on and so forth. And I mean, for this small fragment, you can do it by hand, and for 8 Russulas, you can do it by hand. That seems fine, but when you have 1,000 and the sequence is longer, you have software that does this for you. And what the software does is basically group the specimens based on their DNA sequence. So if you have 1 and 3, these 2 would be in the same group, and then 8 would be in this orange group, and then 5 would be in the yellow group. I got that wrong, didn't I? This should be 5 and this should be 8. Never mind. Okay. So once I had the groups of the specimens, one of the things, the thing to do was to take the information from the database about all the characters and then group them with the DNA-based groups. And what I'm showing you now is each of these pie charts is, on this side is the cap colors. And so this group of Russulas that I called cerulins because they matched the type specimen have these colors of brown for their cap. And so there are different shades of brown, but it's generally a brown mushroom. Russula, what I call Amanita, because again, I could give some names to some of the Russulas. About half of them were yellow, but the other half was red. And so the main thing to take away from this is that you get a general pattern of the colors of the cap. However, there's a lot of overlap between species. And similar story for the spore colors. And so you get species that have paler spores colors and darker spore colors, but again, there's variation within the groups and overlap between the groups. Some of the characters were kind of fun. So ferric sulfate is something that we've known works for the Zarampolina group. The shrimp Russulas is that when you rub the little crystals against the flesh of the mushroom, they turn this sort of greenish color, and that doesn't happen in other ones. So what I'm showing you in this graph here is each species is a row, and the color of the bar chart is showing you what they turn into when the ferric sulfate is rubbed against the flesh. And so these two, Russula viridifosca and Russula rampolina, turn green almost all of the time, while the other ones don't. And other characters were a bit revealing for me because I've always found them a little, sometimes they can help you and sometimes they're just not that great. And this is one of the taste of the gills. I don't know if you've tried to identify Russulas, but one of the characters is how peppery they are. So you put a little piece on your tongue and you wait for it to burn and then you spit it out. And then sometimes it doesn't burn. But some of these groups do show that half of the time they will burn and half of the time they won't. So that was helpful in understanding what kind of variation we would expect from a DNA-based group or a species. It's not perfect for species, but it's pretty close. For the Russulas where I had the most specimens and the most confidence that I was looking at a new species, then I did an extensive microscopy, and so these are some of the drawings I did. These are some of the pictures I took. And so I described 9 new species with the help of Danny Miller and my supervisor. And these were published after we characterized the diversity. What happened with this data was fun for me because it meant that I got to collaborate with people like Danny, who is a champion member of the PSMS. And we discussed at length what sort of thing he could include in his key online. So I think his key is actually really good, not just because he collaborated with me, but I think it's because he really listens to all the data that he can find, and he's very thoughtful about mushroom identification. And then similar thing happened with this book with Kim and Andy. So this is actually number 2 bestseller in British Columbia right now. And this is Kim looking fancy in front of the in Banfield on Vancouver Island. And this is Andy explaining mycorrhizal fungi. So Andy and Zoe are trees, while Cora is a mycorrhizal fungus. And we collaborated, well, I guess they collaborated on the book, but I was happy to help them with trying to describe the variation that you might expect from Russula for the book. Okay, so for this part about Benwu, what I want to, how I want to conclude this part about Benwu is that this is one of the first graphs I ever did after I got the database together. So this is not based on DNA at all. This is just looking at the specimens. And these are the years, all the years that he collected. And this is the number of specimens for each year. The colors of the little bars here, the gray, the darker gray represents the number of specimens that have a species epithet next to them. So that means that Benwu decided to call the specimen Russula cascadensis or Russula adusta. And the light gray is the number of specimens that Benwu decided to just call Russula species. So couldn't assign a real name. And what you would expect from somebody who works on a group for 30 years is that the number of named species would increase because my assumption at least is that you would feel more and more comfortable or confident in what you're seeing. But what you see in this graph is that doesn't happen. And I think that speaks to the honesty with himself about how confident he was that whatever was in the book that had a name didn't actually match what he was seeing in the field. And I always thought I really appreciated that about him. So I guess we can all learn from him in when everybody tells you a name of a mushroom, you can always, if you have a suspicion, maybe it's not just you. Maybe it's truly not the case that we have a name for it. And I wanted to share this little note that he had in one of his collection sheets. I'll read it out for those who need it. So in the remarks section, which is a little section at the bottom of the sheet, he says, "End of film roll ruined by idiot who opened camera before rewinding." And I always, when I saw that, you're entering all these things in a database, and I thought that was the funniest thing. And I, on top of his honesty in naming Russulas, I also appreciated his humor a lot. So I'm happy to take questions about Ben Wu, and I'll move on a little bit about the conservation. I think you can write them in the chat, and then they'll read them to me. So I wanted to show you a couple of pictures of things that I saw in the field in the last couple of days. So I am told this is not a gymnopus anymore, it's a Colivia. But can you see the furry foot? How glorious this is! I've never seen such a great specimen of this. I really like this picture of this fungus a lot, not just because the eggs look cute, but it has this part here where you can see that there's the membrane that covers the bird's nest, but you can still see the gelatinous matrix that that holds the eggs in this bottom one. This is the Nathaniela. There's a beautiful mushroom. And this is the bleeding Mycenae. Okay. So this is a little excerpt from this little mushroom app that UBC put out. And this is a picture from Adolf Cheska. So I haven't seen this mushroom, but I just wanted to make sure that everyone knows that you shouldn't eat this mushroom. People get it confused with the straw mushroom, I am told, which is a mushroom that is edible in Asia, but it's completely different from Amanita phalloides. And others have confused it with puffballs. So when the Amanita is young, it will be inside an egg, and it might look like a puffball, but the only thing that you can do about that, I think, is just cut whatever you think is a puffball in half and make sure that there's not a little tiny deadly Amanita inside. This is one of the mushrooms that we know the most about. I guess its geographical range, its toxins. And what I think is really exceptional about it is that we know how it's spread in California. This is a graph from a paper from Anne Pringle in 2009. And here you can see it spread from 1938 to 2009, or maybe a little earlier than that, maybe 2007. And we know that it's not native. We know it comes with hornbeam. And here for example, in Vancouver, it's found in East Vancouver, and it's found in Victoria, and it's caused some lethal incidents. What is really exceptional about it is the detail with which we were able to track it. And part of it has to do with the fact that it's so deadly. But I wanted to remind you, this is a good segue for this, that for most fungi, the current geographical range is really unknown. We just don't know where they occur. And if we know where they occur, we don't know the edge of their range. And part of it has to do with what I've been talking about with Bengu. So that they're so difficult to identify that we just won't know if we're mistaking it for something else or not. The other side of that is the fact that they have cryptic life cycles. I like this diagram by Karen Hughes and Ron Peterson, where they show you that there's a lot more than just the mushroom itself. There's a whole other part of the life cycle, which is in these mushrooms underground or in wood that we don't know about. What that means is that for 3 to 5 days of the year, maybe more, the mushroom is the way that we see it or the way we identify it. But for the rest of the year, we just won't find it. It's just be underground, but it's still there. And this has implications for conservation. So the idea that we can see mushrooms, and when we don't see them, we can't treat it as a true absence, is a real problem for the first question on this list, because we have a hard time disentangling if something is rare, like truly rare, something that's truly not found everywhere, and something that's undersampled or overlooked. So this is a real issue. Is the population, because this is one of, I don't know if some of you have tried to rank species for conservation, but some of the things that you're asked to do is to show that a population of an organism has gone down, so that it's been reduced over time and now it's reducing. But with fungi, it is very difficult to decide how big is a population if we can't identify them properly, and if we don't know if we've been somewhere, but it was just the wrong time. It was going to fruit the next weekend, and we just didn't know about it. And then we can go on to this more the most existential question of what is a fungal individual. And Anne Pringle, the person who did the work on Amanita phalloides, actually was president of the Mycological Society of America. And one of her plenary talk was about this question, and this is an open question. We're just not used to, as biologists, to think about organisms that are that can come together in the same body and then separate again and not go through a sexual reproduction cycle in the meantime. And it's difficult to untangle, to wrap our heads around that. However, Rewild and IUCN Species Survival Commission, I want to say, have become these organizations that are calling for the recognition of fungi as one of the three kingdoms of life, the 3 Fs, one of my favorite F words, one of them, and critical to protecting and restoring Earth. So on one side, we know that it's really, really difficult to decide that a fungus is endangered or rare. And on the other side, we're starting to recognize that we really, really need to protect them. So this is creating some sort of friction between these two, this need to conserve and this absence of knowledge. On top of those absence of knowledge about the biology and so on, because we need to know the biology of fungi to be able to really decide or identify species that are endangered, there's this added layer of complication. So what I'm showing you here is maps of North America. This is on your left, you can see a map of North America that's not political, and it's just where fungi may be able to live around our disruption, I should say. And on the right, you can see where how conservation is organized, is at the federal level or at the province level or at the state level, but these borders do not match what the species the species range that we see in nature. So this creates a little bit of a difficulty to decide for, I don't know, for Canada, something may be rare, but most of its range is in the United States. And so how does Canada decide that this is an important thing to conserve? And there's different approaches to this. And one of them is to target specific habitats. And this is working under the assumption that if you see a habitat that is shrinking, then you make the idea is that the species living in that habitat are also shrinking in population, that they're endangered because of it. The other way of thinking about it is to go by species and consider how threatened they are. And both of these approaches are valid. The reason why it is difficult to make decision is because ideally what we would do is protect everything. This is the logical step. This is what we should really do. The problem is that we have limited resources, time, money, whatever, and there is a prioritization that needs to happen. And so these ways of looking at it are helpful in trying to figure out what we should prioritize. So this is a map of different forests in Canada. And one of the places that where things are endangered are in this little green bit in Southern Ontario. The Carolinian forest is a very small chunk of Canada. And part of the reason why it's important is because it's so small in Canada, and to keep it part of the Canadian landscape, it needs to be protected. In terms of species, however, if you had something that had most of its range in Canada or all of its range in Canada, then you could argue that in the face of the world, Canada has a responsibility to keep it alive. So both of these things have their own pros and cons. Both of them are valid, and considering them both is really important. One of the cool things, and this is part of the things I learned in this working group that I've been part of in this Canadian conservation of fungi, is that fungi are making huge strides in being recognized in these efforts. So not only the IUCN and so on, but more practically, the wild species reports, which is something that Canada does every 5 or 6 years, has increased is the number of fungal species from 85 in 2015 to 7,000 in, I think, coming pretty soon, I think 2021, 2022, maybe. And what's very cool about it, though, to bring it back to mycological societies, is that a lot of the information about these, the range of the species and occurrence and abundance of the species are from herbaria and mycological societies and mushroom clubs. Like the VMS. So this is the Vancouver Mycological Society. I don't know if you remember a time when there was no masks, but this is from 2017. And these are tables that we would have out, and how important it is for our type of associations to live in order to give us this number. This is also Kevin Trim that I saw on over the weekend, and he had this lovely t-shirt, and I wanted to share it with all of you because it was so good. And so the role of mycological organizations, and this I'm wrapping up now. Like your mushroom club, like every mushroom club, the mushroom clubs have a deep knowledge of local mycotas. And this is key in understanding, describing, categorizing diversity. Mushroom clubs are also the ones that not only describe this diversity, but provide the tools that become the resources, the official resources often to for the states or for the provinces or for the local governments to understand that diversity. People use the keys that your club provides. The clubs often are actually the reference that people use, that the poison control uses to identify mushroom poisonings. And as hopefully I've been able to show you, your work of going on forests and recording your species lists and taking pictures also helps in the conservation of fungi. So if you're, this is another summary. What I've told you is that Benwu collected 1,000 Russulas, and this helped us understand their diversity. Herbaria are an important institution which benefit from collaborating with associations like yours, and that mushroom clubs have immensely contributed to our collective knowledge of fungi. And hopefully you will, I'm pretty sure I'm preaching to the choir, but just to reiterate that even though there's so many challenges about trying to figure out the biology of fungi and their distributions and so on, all these challenges can be met because it's so important to preserve these species. And these are so on here, on the left are the people I want to thank for the Wu collection. These include my supervisor and staff from many herbaria that have helped me, people that have contributed collections, and so many mycological societies that have like given me little pockets of money or invited me to speak and so on. And then here on the right, I put a picture of the working group for the conservation in Canada, and this is the list of their names because it's all been virtual, just like this meeting. And I would especially like to thank you for listening and for doing what you do. I hope you take Chris's invitation to go and do some iNaturalist recording of your local mycota at the weekend. So thanks. I will take questions if you have any. If not, I'll just chat. Chris.
Yes, we have a few questions. I will relay them to you.
Cool.
Can you hear me? I just want to make sure. So,
Yeah.
Well, the first one is from Ann Goddard, and she wants to know what you feel about the phrase "just another Russula."
I love it. I love just another Russula. I love Russulas, but also did a 6-year project on them. So I have a deep admiration for the genus, and at the same time, I think sometimes, oh boy, this is just another Russula. I don't know what it is. So, yeah, I appreciate the question.
I assume you don't like. I think it was David Aurora that said, "Better kicked than picked" about the brevipees. which I
Yes,
would
that's right. That's what he wrote. in
I would
Mushrooms
disagree with that one.
Mystified, and that's pretty. I think that's pretty funny. I appreciate that too.
Okay, so Dylan has a very serious one with a
Cool.
word I just had to look up.
Maybe
what
I'll
was,
have to look it
what-
up too.
I'm sure you'll know what it means. What was the rubric you used to differentiate species in the paper you published describing the nine new species? And did these 9 new species originate from Ben Wu's collection? So rubric I had to look up, and I guess that's like a feature or something.
So I collaborated with people who had described Russulas before. And so I was following sort of their instructions about what sort of things should be in a Russula description. Since I described them, there was a paper that came out in Fungal Diversity that gives you a little bit of a key to how to do that. So I haven't checked if my descriptions follow exactly what they suggest, what other experts suggest, but the idea is that you want to give as much information as you can about the cellular structures of the cap, of the gills, and what else? And so certain chemical reactions. The species described, yes, they were from the Wu collection. So all of the types are specimens that are in the Burke Museum and are in the Wu collection. Thank you. That was a really good question.
Nice. Anna, could I get you to quit sharing your screen so we can see more of you?
Yeah.
There we go. Thank you. Sandy?
Good call.
So I have a question for my husband. I think it's the same question he asks every speaker. and that is, and I don't know if you'll know the answer, does clear cutting affect fungal conservation?
Oh, I don't know about. Yes, I believe it probably does.
Okay.
I don't have the data here with me, but I would assume that clear cutting does a pretty big damage or at least here in around Vancouver for the old growth. There's some fungi that are only found there, and once we get rid of them, that's it for them. So I like the question. I wish I had more precise data to give your husband.
Okay, here's another unusual question.
Cool.
A little bit unusual. Are you hopeful, and do you see it becoming a reality for mycological capitalists, example, Stamets, funding research projects that have no capitalistic outlook? I guess he's saying pure research and ecological purposes rather than I don't know.
Yeah. I never heard this question before. This is a really interesting question. I never thought about it because maybe I don't have that much hope, but maybe I should be excited about more investment into environmental research from these pockets of money out there. I don't know. Do they have an opinion? I'm curious where this is going. This is what we're missing in person. I feel like we could.
Right, and the name of this person is Oregon Original, so not even sure who that is.
Oh.
Anyway,
Well, I could interject here just a moment.
Do it.
It's in the latest issue of Fungi magazine. It was reported that Paul Stamets has endowed a scholarship in Andrew Weil's name.
Oh, cool.
At his institute. So there's hope.
Yeah, that's a great way to funding students is, from experience, is such an honor as a student to get these little pockets of money. So I love it.
So we also have a question from Gypsy Girl. And actually, I think we'll be able to answer that question next month, because her question is about forest fires. What impact do forest fires have on fungal conservation? And Chris, I believe we're having a talk on that next month.
We are. Thea Chesney, with the botanist with the Forest Service in California, is going to discuss that topic next month.
Yeah, I wouldn't know. I'm curious. Can I tune in to hear the?
Sure you can.
I am curious, and I don't think I have a good answer for that question. It is an interesting question, though.
Chris, do you and Molly have any questions? Just curious. Molly usually, I'm asking because Molly usually does.
I was gonna say, I'm surprised Molly doesn't have a question.
So
No questions, but we all wanted to thank you for putting your database on open access and allowing us to peruse your 6 years worth of work at the click of a button. That's just phenomenal.
It's really fun, actually, to look at all the notes as well, because you can open the photographs and the collection sheets. I don't know if it inspires anyone to have better collection sheets. I hope it does.
Peruse your 6 years worth of. So, do we have any more questions, Sandy?
no, I'm not seeing any. I don't know, maybe I missed it. Maybe it was in the graphs. I mean, did you have I got the feeling that those were not 1,000 unique specimens. Is that correct?
They were, yes.
They were 1,000.
Well, or unique as in, no, they were grouped in species. So not 1,000 species. They were, is that what you mean?
Yeah, right. Do you have a count for how many species, unique species there were among those 1,000?
Yes, I counted 72 species.
Oh, okay.
Yeah. And
Okay.
Those would not include things that are as much on the oak forest, as far as I know, based on where he used to collect. But I think it's a good estimate of the diversity here. Yeah.
Okay. We do have a couple more questions. One from Dylan is, can you talk about the distinguishing features of, and this is weird. I noticed you said Russula, and I always say Russula, except when it comes to shrimp Russula.
Oh.
I don't know why.
Okay, no,
I guess it's shrimp Russula.
Is
so
this
the distinguishing features of shrimp Russula?
Oh, yeah. The group shrimp Russulas are
right. It is. I guess they're calling it a group now.
I think there's 3 species. The very red one, I think, is the one that still needs to be described. We call it a Zarampolina, but I don't think it's actually Zarampolina from Europe. And as a group, all of them have a brownish staining when they're bruised. They have a darker spore print, so it's not white. It's a more of a dark cream or almost ochre. They're not spicy unless they're very, very young. When they're very, very young, sometimes they can have a little bit of a peppery taste. And then if you have the ferric sulfate and you rub it on the flesh, they do turn. That's a very, very good character, actually. Yeah, those are the characters that I would use in the field to identify them. And was the Russula or Russula a question, or was that your consideration sending?
Oh,
So
I know you say Russula, right?
yet,
Or not?
I
Or do you say
say,
Russula?
I think you should just, I think like any Latin name from in that scientific name, you should say very fast with tons of confidence. And that's the only way. This is, I can't remember who said that. I stole it from someone.
Right. Yes, so I've heard you say it. Yeah. Oh, pronounce every syllabus and say it with confidence. I've heard that one. So, okay, here's another question. Actually, there's a statement and then a question.
Okay.
The last major change in range of Russulas as documented by Buck, B-U-Y-C-K, was the last ice age. Is climate change affecting Russula species range, or is it too early to tell?
Climate species range. I think we can safely assume that it's affecting the alpine Russulas already, just because of the change in tree line up on mountains. I don't know about lowland species. I would be more worried. I am more worried about Russula species ranges affected by habitat destruction. And of course, the changing climate doesn't help. But that's not just specific to Russula. I think we can make a more general statement about what does it mean in the Pacific Northwest to have hotter, drier, heat-domey summers and very cold, wetter winters. I think that's probably going to not just, I think that's more of a general thing that's going to affect most mushrooms rather than just Russula. Specific to Russula, I would say climate change definitely impacts the ones up in the alpine or in the subalpine.
Okay. Okay, last chance for questions. Oh, I'm getting thank yous.
Thank you everyone for tuning in. And
Yes, and thank you, Dr. How do you say your last name before?
Bazzicalupo.
Bazzicalupo.
Yes.
Thank you so much, Anna. And we will please be in contact with me and let me know where you'd like that honorarium to go for the Mycological Society of America's Diversity Committee, I think.
I think it's an award. I think that'll be really good because the Canadian dollar. Anyway, I think that'll be a good. I'd like it to go there. If that's
excellent.
Possible.
Well, thank you so much, and we hope that someday in the very near future, we'll be out of this COVID pandemic and we'll be able to cross borders and meet in person once again.
Yeah, let's go for 4A. That would be super fun.
That would be lovely. I look forward to